I don't know if you want to know about that. Egypt now is 2 hours to midnight.
Oh,
So,
really?
yeah, we are 8 hours ahead of. They are 8 hours ahead of us.
Oh.
Yeah. So those guys are like worth a good presentation.
Oh, I'm glad that people were waking just to listen to me.
Yeah, they
I
are
don't
good
know if I
guys.
deserve it, but I'm really glad.
Great. So today it's my pleasure to introduce our speaker, Luana Di Campos. Luana is my lab mate, and she is one of the best computational chemists I have ever met. Why I'm saying that? Because Luana has a great ability to connect theory and application in this field. So Luana employs different techniques at the same time to build a very accurate calculations, very solid calculations. So Luana, on one hand, can work with more basic and more common techniques like docking and SAR calculations and in silico prediction. And on another hand, she has a very good capability to work in more advanced techniques like homology modeling, globularity and orbital energy calculations, and MD simulations. Luana graduated from School of Pharmacy, University of Western Nebraska State. And at that time, she did a lot of computational chemistry work. By that, she moved to get her master degree in the lab of Dr. Eduardo Di Mello in the same university. And then she got her master degree specifically in QSAR analysis. Then she moved to our lab. She's pursuing her PhD in both medchem and computational chemistry at the same time. So she has two fundamental projects, one related to COVID-19, it's pure computational work, and the other is related to medicinal chemistry work. She is sharing one project with me to target chlamydia with two different targets. Uh, Thank you so much, Luana, for joining us today. I'm so glad you are here. I know how you are busy these days, so thank you so much. Those guys are so excited and ready for good science. So let's dive right in. So I will turn it back to you, and if you guys have any question, please type it in the chat box or just keep it in mind until the end of the presentation. Thank you so much.
Thank you, Mohammed, for the very nice introduction. Thank you, everyone, for being here today to listen to me, especially now I know that it's very late in Egypt, so I'm really flattered. Okay, so today I'm going to discuss a little bit about a topic that I really like and I'm really interested in, which is computational methods in drug discovery. So my idea for this presentation, it's not to go so deep into the theoretical part behind these methods, but it's to present the methods and to give a general idea how they work, how they can be applied in research projects, especially in drug-related product development. So I try to also insert in the presentation some nice examples of how we can use these methods, how we can combine these methods to in the drug development process. And I also insert some examples of open source softwares that we can use, that I have used or that I know it's reliable to be used in research projects because they are usually presenting different journals specifically related to computational methods, so they are reliable. Because one thing that I learned from working with these in silico assays is that you can, if you have the knowledge and you have the tools, publish a paper from your home. But sometimes you not have access to those softwares because usually very good softwares, very softwares that are incorporate many different methods, they are very expensive, and sometimes we just don't have access or the resource to buy it. So I try to put some examples. Later I can send the presentation to Mohammed. He can send to all of you. So we have the links in the presentation and a brief discussion about these open source softwares. I hope it can be helpful if any of you are interested in start working in silico assays. So let's just start. So the first question that I wanted to discuss with you is the reason why computational methods can be used, or why should I use these methods in drug discovery? So one question that sometimes I heard is, why should I use this? Because lab experiments are so much more reliable, or they can give me better results. So it's not that when we use the computational methods, we are going to do a determination. We do a prediction. Sometimes this prediction is very good. Sometimes these predictions are not so good. But the overall object of using these methods is to accelerate the process, to turn it more, to give results faster, and to make this overall process less expensive. Because as you guys know, the process of drug development is really long, and it's a very complex process. The chance of success is really low. So the idea of using these in silico methods is to try to help make this process a little less complicated, a little less complex. So if you take a look in the average period of time that one drug from its discovery and development takes to go into the market, it's average like from 10 to 70 years. Sometimes it's a little bit faster, sometimes not. But overall, it's a very long time. And also the cost of this overall process, it's around 5 billion. And the chance of success is really low. So we start testing a lot of compounds, thousands of compounds, and then we have a long process until we can optimize our lead compound to get to one drug that enter into clinical trials. And sometimes it goes all the way to the phase 3 and it gets rejected, and the project is withdrawing from the clinical trial. So this process is very complicated, and the reason of using these computational methods is try to make this a little less complicated. So computational methods can help us to answer some questions that sometimes lab experiments cannot. At least the technology that we have now, the lab experiments cannot provide some answers that we might need a better understanding of the process. So, for example, when we use molecular modeling simulations, we are trying to see each atom of the system, how it behaves in relation to the other atoms around. So this process of analyze each atom of the system is not possible with our currently technology like X-ray or cryo-EM. So computational methods can help us to understand some process that we cannot do it in the lab yet. Maybe in some years we will be able to, but with the technology that we have now, we are not able yet. So some questions that the computer can help us to answer. So what is the target proteins that my compounds can bind? My compound can bind only to a specific target, or it will bind to other similar targets. How this process of binding occurs? Can I improve this the affinity of my ligand to the target? How is the shape? How is the pocket of the structure that I want to target? How it looks like? How can I improve my ligand to develop more interactions with this pocket? So all these questions and other questions we can answer with computational methods. And as I said, we use these methods to reduce the time for experiments, to reduce the costs. And another thing that's also very important is to generate less residues. So if you think about to synthesize thousands of compounds and test these compounds, we generate in this process a huge amount of waste. So we can try to use these methods to rationalize the process. Instead of synthesize 1,000 compounds, we can synthesize at maybe 20 or maybe 10, the ones that are computational methods indicate that have better, most like promising activities. So we can rationalize the process. So another question that we can think about is that if the computer-aided drug design area has ever pushed a drug from the from its discovery to the market? And the answer is yes. Sometimes not all the process was all theoretical. Sometimes it's just an accessory tool, but there has been many cases of successful applications of these methods in the drug design, in the drug development process. And I have some examples here, but I would to point especially the antiviral drugs, because the major success of these methods was in this kind of drug, especially after the 80s, with the discovery of HIV virus. So since they discovered the disease, they wanted to have a drug in a really fast way to try to treat this disease, a new disease that they didn't know so much, and they didn't have any drug to treat. So they decided to use these methods to try to push a drug to the market in a faster way. So in that time, the X-ray technology was being also improved, so they could have access to different virus proteins like integrase and proteases of the virus. So many research projects were focused on get these targets, analyze the binding pockets of these targets, and come up with structures that could bind to those targets. So in this process, many antiviral drugs were discovered, like saquinavir, boceprevir, and umprenavir, just some examples, but for example, also haltegravir, which was the first inhibitor of HIV integrase to go to the market. So these are just some examples, but there are more, especially in the antiviral area of research. Also for the flu virus, oseltamivir and zanamivir. These are the two drugs that are approved to use to treat H1N1. Both of these drugs, they have similar mechanisms of action, and they were both discovered using computer-aided drug design. So those are just examples to illustrate that, yes, computer-aided drug design methods have helped the drug development process and have pushed drugs into the market. So another question that sometimes we can ask is that if we need to be a computer expert to use these methods. And the answer is not. No, we don't need to be computer experts. We just need to be to have a basic idea of how these methods work, how I can apply these methods in each situation, how can I interpret the results from these methods. But I don't need to be a computer expert, a programmer to use. But it's a very interdisciplinary activity. So if you have some knowledge about command line or programming, it will help you a lot, especially because sometimes you get errors using the softwares. So if you know a little bit about programming, or if you know a little bit about command line, you can resolve some problems, some errors or bugs that the programs have in a really faster way. So it will help you, but it's not required. But some people are expert on these areas. And in 2 different occasions, the Nobel Prize was awarded to people that work purely in theoretical methods that were adapted to using as a software. So the first case was in 1998, where these 2 scientists, Dr. Kohn and Dr. Popov, they were trying to develop a method, a quantum mechanical method to calculate like electronic properties of atoms, like how can I calculate the HOMO orbital energy or the Lumen orbital age, the energy. At that time, they didn't have any method that could calculate such specific parameters. So they developed the DFT theory, which is until today the best method to calculate electronic properties. It is very complicated method. It's purely physics, but they were able to adapt this, this physical knowledge and put it into a computer so we can calculate. Also in 2013, these three scientists, Dr. Karpus, Levitt, and Waschel, they adapted the molecular dynamics simulation method that was available at the time to combine molecular mechanic properties and quantum mechanics properties to calculate with better accuracy properties from the dynamic simulation trajectory. So they also won the prize. So as you can see, you can be an expert and this will help you, but you are not required to be. So you can use any of these methods that I'm going to talk to you. So where do we start? So actually the answer for these questions is really simple. We only have two options. Or we start using methods that are based on the structure of the target of interest, or we start from the ligand. Because for a molecule to have activity, we need a target that we can modulate the activity so we have any physiological activity that we want. So we can extract information from the target or we can extract information from the ligand. And once we have that, we can go each direction. You can use both groups, techniques that are in both groups, or you can use techniques that are only one group. It will depend on the availability of the targets or the ligands that we know that bind to a specific target. So sometimes we know that a specific protein is related to a physiological process, but we don't have the crystal structure, or we are not able to generate a homology model that is that have the good quality so we can use in our experiments. So in this case, we can use the information that we have from ligands that we know that have a specific activity, but we don't know where it binds. So it will depend on the situation, on what target you are working, or if you know that are ligands that are known to have a specific activity. So it will depend on the situation, but you can use both techniques in both groups together. And I have some examples how you can do that and how to obtain good results. But I'm going to talk specifically of the techniques that are in both groups. I have selected some techniques that I think is more interesting, and I think it's more important in the drug design field. So we are going to start with structure-based drug design methods. So the first one is homology modeling. Homology modeling can be a really helpful tool when you don't have any crystal structure of your target, or when you have a crystal structures for a specific target, but this target's not from the species that you are working, that you need. So it can help you to predict a 3D structure of a protein. To do that, you need two things. You need the amino acid sequence of your protein of interest. You need to know this information, and you need to have a model, a 3D structure already, an X-ray structure or a cryo-EM, so you can generate a 3D model for your protein of interest based on the 3D structures of that model. So the basis for that, for the technique of homology modeling, is two overall statements. The first one is that the amino acid sequence determines the 3D structure. So if I know the 3D, the amino acid sequence, I can predict the 3D structure. And the second base is that the secondary structure of proteins is usually very conserved among homolog proteins. So if you think about in the development process, is much easier to modify specific residues of amino acids to change the sequence instead of changing overall the 3D structures, which is a more complex arrangement of atoms in the space. So even though if my sequence is not totally compatible. For example, if I have 40% of similarity between the amino acid sequence of my target in the base model, I can come up with a good structures because the overall 3D structures is very similar, even though I don't have for example, 90% of similarity in the amino acid sequence. So those are the two bases in which the overall concept of homology model is based on. So what is the steps of this technique? So first, we need to know, as I said, the amino acid sequence. So the first step is a sequence alignment search. So we are going to search in different database, and in this case, each software uses different database to do this search. So we are going to look for sequences that are very similar to my sequence of interest. So the software is going to analyze every sequence in the database and it's going to align. It's going to do a 2D alignment of all the sequences, and it's going to calculate the similarity. Once the software does that, it returns you an output where you can select which sequence you want to continue to use for the next steps. So usually between 35 and 40% of similarity is good, it's acceptable, but of course, if we have a higher level of similarity, it's better. So once we have that, we are going to build up our model, our 3D structures based on that sequence, that model sequence that we have found in the first part of the method. So in this case, when you choose the sequence you want, the software is going to do another search in different database, but now 3D models database, 3D determined database, for example, PDB database or other crystallographic databases. So it's going to search for structures that have 3D similarities with that sequence. Once this is done, the software generates a 3D structure based on that model that the software found in the database and returns you again another output. So it's going to rank all the possible models after generate the 3D structures, and you are going to choose between the different options. So usually we start choosing for the one that have higher similarity, and also there are other metrics that the software returns you, so you can analyze to see which model is better. Once you analyze, you validate your model. So you have your 3D structure from the sequence that you want based on a specific 3D model, and we want to know if this model is valid, if I can trust this model. So you use different parameters. So each software or each server that do homology modeling is going to have different parameters. So there is no like a consensus of what parameters are better, because it varies with the software that you use. So once you analyze these parameters and you check if your model that you choose have values for those parameters that are acceptable, you can use your model. So you can use this model for docking, you can use for different methods, but you need to validate. So usually one parameter that is very used in different softwares is the Hamilton plot, where you can analyze if the angles of the side chains of the residues of amino acids from our 3D predicted model, they fall in predicted values. If many of the residues has angles very different that are not natural for the proteins similar to that protein, you need to start the process again. If you cannot validate your model, you need to go back and you need to select everything again. So it's an iterative process. So you start changing your sequence alignment, we start changing your your 3D model, and you do that until you have a good model. Sometimes you can get a good model, sometimes you cannot. So I have seen papers that have models with very low sequence similarity. They start like 20 or less than 30%, and they have good validations. And sometimes they have like 50 or 60% of similarities in the sequence of amino acids, and they have very low statistics to validate. So it's kind of, it will depend on what type of structure you have. Some structures, some more complex quaternary structures are harder to validate. Sometimes you are working with a protein that have a more less complex structure, so it's easier to validate. So it will depend on what you have. So here I brought some examples of open source softwares to use in the homology modeling process. The first one is Swiss Model, which is from the Swiss Institute of Bioinformatics. So this institute provides a lot of different, not only for homology modeling, but a lot of different tools for molecular modeling, and they are all free, and they are very acceptable as a tool for publication. So if you go to their website, you are going to find many different options to use, and they are all free, and you can use in your research project. Others software is Modeler. So Modeler is a little bit different from Swiss Model. Swiss Model, you use everything over the internet. So you have a web server, and it's much more automated. You just push buttons and interpret the results. And here on Modeler, you have to have a little bit more of informations. It's a little bit harder to deal with this software, but it's also very acceptable. It's another option that is free. The second technique is molecular docking. I'm sure all of you have heard about docking or have done docking before, because it's a technique that is very easy to do, and you can do in your home computer. The theoretical behind it, it's not so complex. So any hardware that you have, you can it can handle docking softwares. And the idea behind it's really simple. So once you have your target and your ligands that you want to screen to see if it binds to your pocket, the software is going to start generating conformations, and it's going to start taking your ligand and trying to insert this ligand in the pocket in different orientations to see what is the best conformation and the best orientations that we can fit this ligand into the pocket. So I think of docking as a hand and glove model. So you have your hand and you have your glove. If you're trying to put your right hand in your left glove, it's not going to fit. It's very similar, but it does not fit well. So you need to try any different conformation and orientation, and until you can get a really a good fitting inside the pocket. So we can use docking in different situations. For example, the most known is the standard docking, when you have your target. So this target can come from the homology modeling that you have done before, and you validate your 3D structure, or you can directly download it from the PDB database. And you can use these 3D structures to analyze many different ligands. If any of this library of ligands, if any of these ligands can fit in the pocket. You can also use the reverse docking. When you do the opposite, you have a ligand and you want to see if this ligand can fit in a specific binding pocket in many different proteins. So this one is more common, and most of the softwares, at least the open source, they are used for this type of docking, but there are some options that can do this reverse docking, but generally those are commercial software. So it's a little bit hard to be able to use because you have the license. You need to buy the license of the software. So what is the components of this technique? It's two components, the search algorithm and the scoring function. So the first component is going to generate the poses. We call poses any orientation and conformation that the doc software gives you. So the software starts to rotate the ligand in any orientation that is possible and trying to fit into the pocket. So it generates many different conformations and orientations inside the pocket. And the other part of the software is the scoring function. So the scoring functions is the way that the software is going to rank these poses, these generated poses, for you to know which one is better. So this ranking occurs based on energy. So usually these scoring functions are based on the sum of all energy terms. So van der Waals interactions, hydrogen bonding, and other electronic interactions. So the software calculates the energy for all this interaction and gives you a sum of that, which is your docking score. So the docking score is a predicted binding affinity. And usually the lowest as you have, the highest stability, and it is usually the most promising ligand in your data set. But we need to take care of this docking score, because it does not mean that you have a really low docking score, which means you have a really promising predicted pose. It will not tell you if this ligand is going to be an inhibitor or if it's going to be an antagonist. It just tells you that it fits well in the pocket and it's stable in the pocket according to this scoring function. So we need to take really care of the conclusions that we assume based on this docking score. Another important feature in docking is the way the software generates the poses. So the search algorithm that the software used to generate this pose, you need to know, because the conclusions that you are going to do when you finish your research is going to depend on this characteristic. So overall, there are two ways for the software to generate the poses. Or it generates as rigid poses, or it generates as considering the ligand and the protein as flexible. So as you can imagine, the computational demand for the rigid docking is lower, because we don't need to account for every rotable bond in the protein. Usually the ligand, most of the software consider the ligand flexible, so it considers all the possibility for rotations around the rotable bonds for the ligand, but not for the protein. And for some softwares, they can consider some flexibility of the protein. So it can consider the flexibility of the side chains inside the binding pocket, but it does not consider the flexibility of the protein as an overall structure. So depending that, if the software does not consider the flexibility, we cannot account for some compounds that after it binds, it can determine movements in the structures to accommodate it better. So those ligands will not be selected for this kind of docking. So we have two models, lock and key and induced fit. Induced fit is in most of the cases are better, because it can account for these ligands that as an initial assessment, it would not bind, but when we consider this flexibility of the binding pocket, the binding pocket can be adjusted to the ligand fit better. So we need to take care, and we need to search for this in the software documentation that we have, what is the search algorithm that the software we are using employed to generate these poses. We can do directed docking, or we can do blind docking. So there is a difference. When you know the binding pocket, where specifically your series of ligand bind, you do the docking specifically in that position. Don't need to consider the entire protein. You go directly to that position. And sometimes you don't know that information, so you do blind docking, which is you take the entire protein and you ask the software to do the search in the entire protein. It takes more time and are less reliable, this kind of blind docking, but sometimes you have no other option. Another thing that is important for docking is you need to prepare the ligand. So you need to assign the protonation states for the ligand and for the protein, because when you download a structure from the PDB, you don't have hydrogens, you don't know the information for the protonation state, and sometimes you even have missing parts of the protein, sometimes parts that are very flexible in the protein. The X-ray technology cannot distinguish very well, so sometimes you have parts that are missing, so you need to account for that and correct that those problems. You need to assign partial charts, because if you don't do that, you are going to have not accurate scores, because the software cannot find the electrostatic interactions. And then you proceed with the docking, you generate the poses, you score, and then you can do you can interpret those results and make conclusions. Here I have a really quick video to exemplify how docking works. So we will start with your protein. It might be an X-ray file that you have downloaded from the PDB, or it might be your homology modeling after validation. So you know, in this case, they knew the target, the binding pocket. So we start generate the poses. So the ligands are rotated and are trying to fit in any different orientation and trying to make the rotable bonds very flexible and assume all the conformations that it can. When it finds a better orientation inside the pocket, we can analyze that pose and we can start to see if we can increase those interactions. So we can modify the ligand to try to increase. So here we have a pocket that we have space to grow. So we modify and we analyze the score. So if you take a look here, the score is going down, which means it's more stable and makes more interactions. So that's a really fast example of how the overall process happens. So here I just wanted to reinforce that it's very important to take care of the structure and ligand preparation, because some people might think that docking is just taking a protein from the PDB database, inserting your software, and that's it. Let the software do everything. It's not quite that. You need to know a lot of information before you start doing the docking. Like, for example, you can download your protein of interest from the PDB database, and this protein can have a mutation right in your binding pocket. And if you do your docking with this mutated protein, your docking results will not be valid. So it will be really bad if you discover that after you do all the docking and all the interpretation. So take care to always prepare well your protein and to know as much as you can about your ligands in your proteins. So how we can analyze the performance of our docking. So usually we do the redocking protocol. So the redocking, it's a really easy to understand concept. So when you have a ligand that is crystallized in your binding pocket, you can extract this ligand. You can insert this ligand that you extracted from the crystal structure inside your docking run. You do the docking, and after you analyze the predicted pose from the docking and you compare with the crystal structure. So for example, in this case, we can see that overall, the predicted binding pose, which is in pink, it's very similar to the crystallized. We have some differences that we can calculate this difference in position of the atoms, but overall it can put the right groups in the right position inside the binding pocket. So we can say that this is a good indication that our docking protocol is good to predict poses and orientations of ligands inside this binding pocket. So there is a value for this error, for this differences in between the predicted and the crystal structure, and we calculate that using the root mean square deviation of atomic position. For small molecules, usually this value, this deviation, this error, it needs to be less than 2 angstrom. Because if it's higher, it means that the docking protocol is just not good in generate very close to native conformations from ligands inside this pocket. So as you can imagine, when we increase the complexity of the structure, when you have more rotable bonds, when your docking your ligand have more complex groups, this error is going to increase. So usually with little molecules like this one, the predictions are very good. But as you work with more complex structures, the error is going to increase. So for peptides, we can accept until 4 angstrom of deviation because we have many different atoms, and in the RMSD calculation, we are going to account for all the difference from all atoms. Sometimes you can use this redocking protocol because you have a crystal ligand, a crystallized ligand. Sometimes you cannot. So you need to take care sometimes when you read some docking from results from a article, because sometimes they just put a ligand inside a pocket and the software generate a score for these structures, and they say, oh, this ligand binds well to this pocket. But you can put whatever ligand inside the pocket and ask for the software to calculate. The software will calculate and will give you some results. But that does not mean that this specific ligand is going to bind. It's just that the software do that for any kind of ligand that you put inside. So you need to be very careful how to interpret the results of docking. And there are other problems with docking also. As I said, some softwares have limited flexibility. We are not accounting for solvent in the system, so usually we remove the waters from the structures, and we do kind of a vacuum complex. So we take the protein, we take the ligand, we take every solvent, we take every other ion that might be present, and you do the docking. So it's kind of a vacuum. So it does not represent very well the system, the real systems, but it's a way for you to analyze thousands of compounds in a really fast way. So it has its advantage, but it also has disadvantages. And sometimes, as I said, you have a promising score in the docking, but you cannot drive conclusions from that if the compound is going to be an inhibitor or an agonist. You don't have much more information. Sometimes when docking is very well applied, you can have around 92% of accuracy. Sometimes you have 0%. As I said, it's going to depend on how you work with your complex. It's going to depend how much information you had from those ligands and from those proteins that you are working, but you need to understand what you can do and what you cannot do with docking. So other problem with docking, because it does not account for water and for flexibility of the protein. Sometimes you do a docking, you have really good score, the ligand fits well in the pocket. And when you do the simulation, we are going to talk about simulation in a minute, but when you put real systems, and when you put water, when you put ions, when you put a membrane, because your protein might be a membrane protein, what happens is that the binding of the ligand and the complex is not very stable. And what happens is that the ligand goes out of the pocket and it prefers to interact with the solvent than interact with your binding pocket. So sometimes we have really good docking, but you have really low results in the MD. So you need to know those differences. So overall, MD is a much more reliable, a much more better representation of the physiological systems, because you can account for the water's presence in the real system. You can analyze how the protein interacts with your membrane. So in this case, I have in green, I have the membrane protein. Here we have the membrane, and around you have water. It is not possible to see here, but we also have salts like that we usually have in the physiological conditions like sodium chloride and so on. So we have a better descriptions of real systems. So here I have an example of docking. So here it's really nice, this example, because this is a compound that came from discovery until clinical trials in less than 2 years. So it's a really improvement of those 10 to 70 years that we saw in the beginning of the presentation. And they only had to synthesize 31 compounds. So they didn't have the crystal structures, but they built up the homology model of the receptor. And once they have this model, they used DOC to screen more than 40,000 compounds. Once they analyzed this amount of compounds, they have selected from the docking score 78 compounds that had good predicted docking score. They bought this 78. They analyzed how well these compounds inhibited the receptor. And once they found one ligand that inhibited the receptor with good IC50, they started to work on this best compound as a lead. So they analyzed how this compound bound in the binding pocket, and they started to see how they can improve this interaction. So they saw that in the binding pocket we have still room for more substitutions to expand this ligand to reach more parts of this binding pocket. So this is the lead compound, and this is the overall base modifications that they did from this lead compound. So we can see that they removed this group here because it's kind of close. It's to this part, so they remove the methox groups from here, and they inserted groups in this part to fit this part of the binding pocket, and they continue to optimize until they get this naloxone structure. So they did all of this in a really fast way, and they pushed this drug to clinical trials phase 1 up to 3 in a really fast way. So it's of how can you optimize the process, reduce the time, reduce this experiment, and have good results. Here I have some examples of open source softwares. It's interesting because in the case of docking, the software that is most cited, that's most used in publications are a open source software, which is Autodoc. So much of the majority of publications that use docking use Autodoc. So it's interesting because it's a open source. And I have other examples, a different methodology. I try to put softwares with different algorithms of search. So we have options. So I will send you the presentation so you can take a look better on these softwares. The other method that I want to discuss is molecular dynamics simulation. So what is this? It's a computer simulation that can analyze the physical movement of atoms and molecules. So we can analyze atom by atom in the system. And we can, once we know the position, the initial position of each atom, we can predict how all the atoms in the system is going to behave. So if you take a look here, you can see that this atom can interact with the other atoms and how the other atoms respond to that interactions. So you can see here that it comes, interacts, and it's not bounced back. And it keeps interacting because we have attraction forces and repulsion forces that it's going to make the interaction profile different from each atom in the system. So how we can know that? It's a very complicated theoretical, there's a very complicated theoretical behind it, but the overall idea is very straightforward. So if we know the initial positions of each atom in the system, we can apply the Newton's laws of motion to predict over the time where each atom is going to be as the time goes by. And we can also predict this position of the atoms because we can consider the electrostatic interaction. So it's not only the forces, but it's from the one atom being pushed from other atom, but we are also considering the repulsions and the attractions forces that are exerted into each atom. So it's really interesting. I'm not going to go into detail, but the overall idea is this: it's very straightforward. I think of all these methods that I'm discussing here, this one is the most nice one, and it's the one that the reviewers and the journals are looking for to see in your paper. So if you see here, I just make a quick search in Scopus, and you can see here from 1972 up to 2020 how much we have the increase in publications using this method. And it's not for drug discovery. This method is using many different areas of research, and it's very well accepted, and the reviews really look forward to see this in your paper. So what informations can MD provide? Many different informations. For example, we can analyze conformational changes. So when you have a crystal structure, you have a photograph of the structures in one conformation, but that does not mean that the structure is going to be all the time in this specific conformation. So with crystal structure, we cannot have the overall idea of how this biomolecule behaves over the time. So for example, we cannot analyze how, for example, when a ligand binds to its receptor, how it's the changes that happens in the receptor, how all the interactions are performed during this binding process, and how the changes in the receptor occur. So with MD, we can do that. We can predict how the system is going to behave. So we can also predict how a protein fold. So this is very used in, for example, the research to understand how Alzheimer's and Parkinson's disease are developed, because the process of Alzheimer's and Parkinson are related to proteins that fold in different conformations. It not folds in the right conformation and starts to aggregate and generate the disease. So MD are very helpful in this case, because since these molecules are very, they move very fast, much faster than normal proteins. X-ray cannot give so much information, but MD can. We need to consider the time scale of the process, the physiological process that you are studying. What is the time that it takes to happen? Because you need to simulate the system for as long as the physiological process would take in real life. So for some analysis, it's really fast. For example, a bone stretch would happen in femtoseconds, but for example, allostatic transition in a protein would happen in milliseconds. So as much more time the process takes to happen in real life, as much more complex and much more computational demanding it's going to be to simulate it. So if you have very complex systems like proteins, membrane proteins, you are going to have a more complex system with more atoms, and the simulation is going to take more time to do it. Usually we do not simulate this type of systems in our computer, because it does not have the right hardware to do it. It would take years to finish. We do it in supercomputer. So for example, here we have access to Holland supercomputer that we can send our analysis to be done there. So usually for example, in an allostatic change, if we would do this with our normal computer, a regular computer, it would take 30 years. But with the supercomputers, we can do it 3 days. So it's really faster. Regarding the steps of the MD simulation, we have our protein. We prepare our protein. we assemble this, the overall structure. So if it is a membrane protein, we need to model the membrane, we need to model the protein, the ligand, and the solvent around. Sometimes we need to add counter ions to neutralize the charge of the protein, but we assemble everything, and then we can simulate for as long as the physiological process that we are interested to observe, how long it takes, we need to simulate as would happen in real life. And then we can analyze our results. One important thing is to consider how we are going to treat our conditions in the system, because, for example, for ligand binding complex, we let the temperature and the pressure constant over the entire time of the simulation. But sometimes we want to study how a change in temperature can affect the folding of a protein. So in this case, we would have to use a different set of different way how to consider the temperature and the pressure. And sometimes we also need to consider changes in volume. So this is going to depend on which system you are using, and also what are you want to study from that. So you need to know all these variables to put a complex that it's very similar to what we have in real life. So here I have an example. So this is from our study with COVID. So the idea behind this study is that we wanted to mimic what happens when the COVID interacts with its receptor. So here in yellow, we have the receptor binding domain of the virus, and here we have the host receptor. So the part that interacts is really one alpha helix from the structure. So we analyzed that, and we thought maybe we could come up with a peptide that mimics this alpha helix and conserves the 3D structure, because we can staple these peptides to conserve this alpha helicity. So would be efficient to block the virus to interact with its receptor in the host cell and to be internalized. So we come up with different peptides, we modeled, we optimized these peptides, we did the docking, and from the docking we found that this structure had the better scores. After analyzed the pocket and the RMSD, we saw that it was a good prediction. So we used MED to analyze if in real systems this stapled structure could maintain the alpha helicity and interact with the receptor binding domain. So let me see if I can, I don't know if I can. So I have a video that I'm going to show. So here we have our peptide. It has some variations in the secondary structures, but after equilibration, you guys can see that it can conserve its alpha helicity most of the time. So after a period of equilibration, it conserves and interacts with the receptor binding domain, because it can conserve the 3D structure. So this is the control. You are going to see that that is not stapled, it's not locked in an alpha helicity. It's going to start losing its alpha helicity over the time. So can you see it's going to unfold and loses the ability to interact with the receptor binding domain. So those are results from our simulation. After analyzing these results, we also analyzed some metrics to see if our simulation had equilibrated, if everything was okay. And we see that, yes, the fact that our peptides have this stapled, it allows to interact with the receptor binding domain because it conserves the alpha helicity over the time. These are some examples of open source softwares. We have Gromax. Gromax is one of the most used. It's free. The only problem with these two softwares, actually, both are free. They have different algorithms, but overall they are very good in predicting when used in the MD simulations, but the problem with this tool is that they are command line. So if you don't know how to use command line, you are not be able to use these programs. And they have command line interfaces because they are software developed to be very demanding regarding the computational the calculations that is going to be done in the computer. So when we don't have a graphical user interface, we reduce this computational demanding from the hardware of our computers. So it helps to make the process even faster. And the other part is that usually the supercomputers, we cannot connect to those supercomputers, those servers, using graphical using interfaces. We need to contact those servers using command line. So as I said, you don't need to be an expert, but you have to have some basic knowledge of how to use command line. So the second group is the ligand-based drug design. So when you don't have the target, or when you don't have a good homology modeling, you can use these methods. So the first one is pharmacophore modeling. So you can use the information from our known ligands. We know that a specific ligand can bind to a specific pocket, so we can extract from the ligand this information. So we can build up a 3D pharmacophore, and we can use this 3D, for example, in virtual screenings. So how we generate these pharmacophores? So we just take some compounds that have similar structures, and we know that it binds to a specific pocket. So we align all these ligands, and the software screens this alignment to see which characteristics appears in every ligand. So once these characteristics were screened, it gives you all the characteristics that were selected from this alignment, and in the 3D space, what is the distance between each characteristic, and we can use this to search for new ligands that can fit in this model. So sometimes you can get luck, and you can get a after your virtual screening using pharmacophore modeling, you can come up with structures that have the same pharmacophore, but is a very different structure. So when you case, for example, if you want to work with a specific structure, but that structure has a patent, you can use this pharmacophore modeling to try to come up with new structures that conserve this pharmacophore, but have different, very different structures. So it's a really good way to do virtual screening, especially if you are going to use a large database. So these are an example of how we can use pharmacophore modeling. So in this study, they started from a data set of 42 compounds that they knew had antimalarial activity. So they used the pharmacophore modeling. So they come up with this pharmacophore. So it had aromatic characteristics, and also it had hydrogen bond donor and hydrogen bond acceptor. So they screened the data, a database. So they screened the zinc database. They come up with these different structures, and they use QSAR model to predict if these structures that had the same pharmacophore, what it would be the antimalarial activity. So they wanted to know what is the specifically IC50 for these structures that were found in the database. And they found that the best structure had a better activity, better IC50 profile comparing to the initial structure. So sometimes you have good results, and you can apply other techniques like QSAR model and also pharmacophore. Sometimes you cannot. So sometimes you just really need to have a little bit of luck. So here I have some options for, I don't know why this is happening, but anyway, it's a mistake. So here I have some options of pharmacophore modeling open source tools. I have two. The first one is PharmaGist. which you can use to generate the pharmacophore model to do the virtual screening. The second is PharmMapper. So in this one, we are going to do the virtual screening in the opposite way. Instead of trying to find new ligands that fit the binding pocket, we are going to try to find proteins that can have pockets that our ligand can bind to this specific pocket. So the virtual screening is a very nice way to do different search. So you can search for new targets or you can search for new ligands. So here I have one example of each. As I said, virtual screen can be used for different purposes. So instead of doing a screening in the lab, we can do this in the virtual environment and try to come up with a low number of compounds that from our methods, it has better probability of binding to a specific target or to have a specific activity. So for example, in this study, they used this a database that they download from the Zinc database, and they applied both the pharmacophore virtual screening and also docking. So from the results that they had from the virtual screening using pharmacophore modeling, they filtered that results with docking, and then they come up with a smaller number of compounds. So usually you start with thousands of compounds, so you can use different methods to try to filter and to reduce the number of compounds. So you can use pharmacophore, you can use QSAR, you can use docking, and you also can filter using drug-like parameters like Lipinsk, rule of 5, or other parameters specific for the type of target that you are working. Another thing that I wanted to point out is that when you are working with virtual screening, one really nice tool that you have is Zinc database. So Zinc database is a free database that you can download the entire set of compounds. It's almost 1 billion of compounds. Some are available to buy, others are from research, but you can download the entire database. In this database, come already prepared for docking. So you don't need to do that step before docking of preparation of this ligand to add the protonation states or to add the charts. It comes ready. So you download and you can do directly, for example, a virtual screening using docking. So it's a really nice tool that we have for free. The other part that I wanted to discuss regarding ligand-based drug design, it's QSAR, it's quantitative structure-activity relationship. So this model, this method creates a mathematical model that can predict activities of compounds that do not exist yet. So based on the set of compounds that exist and were tested, we can extract information to predict activity from something that does not exist yet. So it's not magic, it's not science fiction, it's just based in science validated methods. So it's really nice because any other method that I showed you can predict exactly what is going to be the IC50 of your molecule that does not exist yet. So QSAR can give you, this molecule is going to have an IC50 of 5 micromolar, for example. It is 100 precise? No. Sometimes it might be, you predict 5, but it might be 5.3 or 5.9, but it's going to be close. If your model is good and validated, it's going to be close. So it's really nice because you can say, you can give a number, how much activity my compound is going to be. So I synthesize only those compounds that from my prediction, they have good IC50s value. So it's really interesting in the number of publications it's in, from the development of those methods until now, it increased. Sometimes we have some variation, but in 2020 we have the highest number of publications. So it's a very nice tool that we can use. So this QSAR model, it's trying to correlate the structural features of these compounds with the biological activity. So this method tries to extract it from the ligands that we know that have activity. What is the chemical structures that, what is the chemical groups or the chemical features that make this compound have activity? And we use that information to predict the activity of something that, of a molecule that does not exist yet. So it's a mathematical relationship that can correlate chemical structure with biological activity. So the method that are behind, the theoretical part behind the QSAR is it's a lot of mathematical concepts, but the idea is really easy to understand. So if you take a look here, we have different pumpkins. We have some smaller pumpkins, we have some bigger pumpkins, some have a specific shape, some have other, some have a higher weight, some not. So we have different possibility, and all these are pumpkins. But this one is a very specific pumpkin. This is a Halloween pumpkin. So if I want to describe this as a Halloween pumpkin, what I would tell you, what are the features in this pumpkin that I would describe to make a person to understand that this is a Halloween pumpkin? So I could say, oh, this pumpkin has a eye-shaped cut, it has a nose-shaped cut. So I would describe everything that is important to this pumpkin to have a specific activity. So with QSAR, it's the same thing, but the features is chemical features that we need to extract from the ligands, and the activity is a biological activity. For example, antimalarial activity or any other activity, or anti-cancer, or even toxicity. We can use QSAR to predict toxicity. So for example, if I want to analyze these compounds and say if these are genotoxic or not compounds, I would look for structures in these specific groups in this structure that could have genotoxic effect. So QSAR does the same thing. It screens the compound and tries to extract the information that is associated with the biological activity. So the general approach, we start from known ligands. So we need to have a data set that we can analyze, that we know has a specific activity. So once you have this data set, we are going to extract information from it. So one important thing is that we need to have a good amount of data. So we have, generally if we have 30, 35 compounds, it's enough sometimes. But if you have more compounds, you have more information, more chemical information, and probably your models is going to be better, have better predictivity. So as more as better. So you analyze this and you generate molecular descriptors. So molecular descriptors are chemical information in form of numbers. So once you have that numbers, that information that we extracted, we are going to correlate this chemical information with the specific response that we are analyzing. For example, anti-cancer activity. Once you have that, we generate the models. So we use mathematical models to build up this QSAR model. So we use like linear regression or any other type of mathematical strategy to be able to correlate the information that we have with the activity. Once we have the model, we can validate and we can use to predict the activity of other compounds that can have the same activity as our initial compound. So at the end, a QSAR is going to give me predictions, and the ideal word is that my prediction is very close to the real activity. So for example, if we analyze all the activities, we can see that, for example, this one, it's a very good prediction because it can predict almost the same number as the real activity. For some other compounds, we have like higher error, but we expected it. We expect that we have errors, and these errors be distributed along the entire range of activity. So this is very good. For example, this one is a very good QSAR model. So regarding the chemical descriptors, the principal component of QSAR are the chemical descriptors. So chemical descriptors are chemical characteristics, features that we can use the computer to analyze and to convert in numbers. So these numbers we can correlate with the value of biological activity, and we generate a model. So a model is a mathematical equation that we use to predict. So we have different class of descriptors. We have 0D until 4D. So 0D are descriptors, for example, number of atoms in a specific structure. That would be a 0D descriptor, because it does not give any information about the shape of this structure or the 3D profile of this structure. It just gives me the number of atoms. So it would be a 0D. 1D would give me information about the groups that are present in this specific structure. 2D would start to correlate the groups in the position that it appears in the structures. For example, the number of carbon and nitrogen that are present in this specific structure with a distance of 7 bonds. So this would start giving me some information of how this molecule looks like. 3D, we start have information about the 3D shape of our molecule, and 4D we have another dimension would be the time. So we need to consider the time in our calculation, because a molecule is not a rigid structure. It can rotate, it can have flexibility, so we need to consider the time, because as the time goes by, the structure assumes different conformations. So once we extract that information and we put that information as number, we can correlate with the activity. So we use those different algorithms. Like I said, a linear regression, we can use machine learn, you can use principal component analysis. There are thousands of those methods that use to correlate the information from the structure in the number of values and the number of numbers with the activity to generate the models. So we have there are other types of QSAR, but the most common ones is 2D, 3D, and 4D. They are similar in the concept, but how they generate the data is different. So 2D, a 2D QSAR model would have this form, a mathematical regression, nothing more. It's an equation that we can use to predict. A 3D would give me, it also is a mathematical equation, but the equation is hidden in a 3D model. So this would be a 3D QSAR. So these points, for example, this one in blue that is that has a square shape, this would be thousands of descriptors in this position. So we would not be able to put thousands of descriptors in representing the mathematical equation. So we represent as 3D because it's also easy to analyze and to interpret. For example, if I say to a medicinal chemist that I need a compound that have a higher number of carbon and nitrogen at a distance of 7 bonds, it would not be very easy for this medicinal chemist to come up with structures, because there's no much information. But if I say, oh, in this position, I want a bulkier group, because here I can have more hydrophobic interactions in this part. So it would be easier for the medicinal chemist to modify the structures. So these are the little differences between the QSAR, and I think that 3D QSAR and 4D QSAR is much more easier to interpret, and it's more applicable in real life situations. So the only difference between 3D and 4D is that here in 3D, I use only one conformation from each compound. Usually it's the most stable conformation. But as I said, the molecule acquire different conformations over the time. So if we want to consider that, we used to use 4D QSAR, which I would use many different conformations generated from the MED. And for each compound, I would make, sometimes it's thousands of conformations, and I extract the descriptors from these thousands of conformations, and I generate my model so I can account for information from different conformations in my compound. So overall, I need to have the compounds. In 4D, in 3D, I need to align these compounds so I can start to see where I have different groups. So I start to have information of how I modify what would happen with my activity. And then I can start generate the descriptors and have my final models. So I have worked with 2D QSAR, and it's a really nice way to correlate chemical information with activity, but it's really hard to interpret and it's not very applicable in real life situations. So if you want to work with QSAR, I would recommend 3D and 4D. So here I have an example. So in this paper, they use 26 derivatives. They used a 3D method. So they align the compounds and then they generate the descriptors. So as I said, each of these colored different shape positions, they are thousands of descriptors in this in every specific position. So for example, green in this position would mean that if I have any bulkier group, I would have a increase in the activity because hysterically in this position it's favored, and so on. For example, blue would tell me that in this position, in more negative charge would be disfavored, so the activity would go down. So we can analyze the result, we can use to predict the activity, and we can also use to generate new, to design new compounds using the information from this QSAR, and we can know what would be, we can predict the activity of these new compounds, and we can see if we do a good QSAR and we extract all the information from the model, we can come up with really better compounds. For example, in this study, they come up with this 35 compound, which they tested. They predicted that the PIC50 would be 5.51, and the actually measure was 5.03. It's not the same, but it's really close. And they tested and they saw that this compound had an IC50 3.1 micromolar, comparing to the control, which is lamivudine. We can see that the IC50 for this compound is way higher. So this was a really successful application of those methods. So I have here some open source softwares. Here I have one for 3D, one for 2D. This one is also for 2D, and this one is for 4D. All of them are open source. The last part is some properties predictions. So as you guys know, sometimes we have a very nice compound with good activity, but it fails in the pharmacokinetic profiles. So we have some methods that we can try to predict some descriptors that can help us to distinguish between good promising compounds regarding to these parameters and bad compounds. So for example, using this software, which is Osiris, the name, we can analyze different properties. For example, the compound can, the software can give me information about if this compound is going to be mutagenic or tumorigenic or irritant. These predictions are based on fragments in the structures. So when I input the molecule, the software is going to screen all the fragments present in the structure. So it's going to fragment these structures in many different parts, and it's going to analyze and compare with the database. So for example, if I have an epoxide in my structure and the software finds it and compares with the database, it is going to give you a sign that this fragment is associated with genotox effect. So it's just as a screening. It's a fragmentation of the compound, comparison of these fragments with the database, and it returns you an output with specific information. We have some other drug score and drug-like scores. So each software is going to give you different information. This one, it's nice because it's free, can give you some drug score parameters. Other one that I really like is another from the Swiss Institute of Bioinformatics, Swiss ADME, because it gives a lot of different parameters. For example, it can give you some parameters if the molecule is going to be good GI absorption, or it can give you parameters if this molecule is going to be, it's going to have high probability to cross the blood-brain barrier. So it gives a lot of different parameters that you can analyze in your molecules. Here, last, I have these helpful tools. So this is a screenshot from the Swiss Institute of Bioinformatics website. So if you want to look for any tools that you want to use in your research, you come to this link and they will have some different groups here. So for example, if I want to look for QSAR tools, I can click here and I will have a list with a brief descriptions and links to take me to the website of these softwares, and I can use this to, if I want to do any specific study and I want a tool to do that, I can come to this page and they will provide me some options. With that, I finish. It took more time than I expected, so I appreciate your time and your patience. I would like to say thank you for all of you, and especially to Mohammed for inviting me to be here today. I leave you with two pictures of my home country, which is Brazil. The first one is Rio de Janeiro, which is a very beautiful city. This one is Iguaçu Falls. So this was close to the city that I used to live before coming here. It's a very beautiful place. So if you guys any day have interest in to go to Brazil, I would recommend these two places. So with that, thank you, obrigada, which is thank you in Portuguese. I really appreciate it to be here today.
Thank you so much, Lorena, for such a great presentation. Brilliant as usual. So guys, do you have any questions?
Yes, please, Doctor.
Sure.
First of all, I'd like to thank you for this fruitful presentation. It's condensed specifically for those who don't have any experience. Well, secondly, my internet connection was not stable while you were explaining the paper regarding MD simulation about COVID-19. So I'd like to ask, the project aim was to, to preserve the conserved region of the alpha helix of ACE2 receptor, or what?
Yes, so by some crystallographic studies, they knew that the part that makes the COVID to be able to interact with the receptor is this alpha-1 helix. So we have the overall receptor, but the part that really matters for the interaction is this alpha-1 helix. So we cannot take the entire alpha-1 helix and put as a drug, for example, because when it goes to the body, it's going to lose this alpha helix conformation and will not be able to interact. So we thought that maybe if you can take only the specific small part from this helix that have the right residues that interact with the binding domain from the virus, and when you use this staple, we can lock the conformation as a alpha helix. So our hypothesis that when you put in an experiment, the alpha helix would be maintained. So with this 2D structure in position, in the right position, we would have interaction with the binding pocket. So we hypothesize that if we change also some groups to increase the affinity, this would interact with the receptor binding domain of the virus and not the receptor in the cell. So we would like to close all those parts in the virus that interact with the host cell to prevent this virus to enter inside the cell. But we didn't know if we could, with using this staple technique, if this would maintain the alpha helicity, or when you put with water and salt, it would lose its right conformation. And from the MED, we can see that one of our staple, we have 18, but only one could maintain the right conformation to interact. And comparing to the control loses the alpha helicity and cannot interact. So it goes out from the pocket and interact better with the solvent, the control. And this not happens with the R modified peptide.
Okay, thank you so much.
So we have one question in the chat box. Could you please see it?
Please, please.
Okay, so if I understand it correctly, it's asking why not have web servers instead of a graphical user interface. Is that correct?
Ahmed or Mohammed?
Can you hear me now?
Yes.
Okay, so my question lines in MD simulation, if we want to reduce the software to consume more, computer CPUs and RAMs or whatever, why don't we make it an online server that would be more accessible and would reduce this consuming?
Okay, so regarding the software, it's not possible to, at least now, to do it because we can use software online, but the calculations, the server that is going to do, it's really not as a web server. So it's a server that it's from a organization or a university, so it's very expensive, and they usually have those you can access via internet, but they are not accessible to the public. So it's not a web server that we can go to the internet and use as anyone could. So because it's very expensive, those services, I mean, the structure of the cluster of computers, so someone needs to pay for it, and are usually big universities or big organizations. So they would not make profits if they take this and make available to everyone. And if they do that, these calculations are so heavy that they would overload the system. So they need to control how many people are using the system at the same time. So this is the major problem. The software could be made available, but the server, the cluster of computers behind to do the calculations, to run the software in, usually they cannot be online because of those problems, especially overloading the system with many calculations running at the same time. We would have to have a super higher, a lot of cluster of computers to be able to do that. As you can imagine, for example, when you have a website, when you have too many people accessing it at the same time, the system goes down because it cannot run with too many people accessing it at the same time. The same concept could be applied to the supercomputer. So I think the major problem is that.
Okay, thank you. I have another question. If you
okay, would be the last one. Go ahead.
Okay. Why don't we use docking analysis if we want to reduce the time regarding to the MD simulation? If we want to know, if we want to limit the conformation of a docking molecule after making docking, then use this information to and apply it in MD simulation so we can reduce the time. Is that possible or not?
Yes, actually, when at least in drug discovery, usually we use MD after we have filtered our initial data set using docking. So for example, in my study, I had 18 peptides. I didn't do MD for the 18. I used docking, and I did MD for only the 3 best scoring peptides from the docking. So the docking is a very good in ranking compounds and rationalize the process. So instead of doing MD for 18, I did only for 3 because the docking told me, oh, these 3 have better chances of interacting with the binding target. So usually you use docking as a filter tool. Instead, with virtual screening, the same thing. You use docking to reduce the number of compounds, and then you synthesize only the ones that the docking provide good results. and you reduce the number of synthesis, or you reduce the number of MD runnings, because for some MDs, it takes a lot of time, like 1 month, 2 months, depending on what biological process you want to study. So yes, that is actually the idea of docking, to rank compounds, to rationalize the process.
Okay, that's great. So it's 2:30, I think. So thank you so much, Luana, for being with us today. I think if we last more than that, Aramis may be mad at me. So Tahir,
No.
thank you so much.
I appreciate very much. It was really nice to talk. It's a topic that I really like and I get really excited to talk about. It took more time than I expected and I predicted. I hope you guys can forgive me. And if you have any other questions, you can ask Mohammed for my email or any other help. I'm glad to help in any way I can.
Great, great. Thank you so much. So see you then. Bye. Bye, guys. See you next week.
Thank you. Bye-bye.
Bye.
Thank you. Bye.